Thank you very much. I'd like to thank Esper and all the meeting organizers for inviting me. It's been really wonderful, just as it was last year. I especially enjoyed the lunch with 8 of the students yesterday, and to capture their enthusiasm, it was really, really fun. Today, this morning, I'm going to be talking about long-term delivery of monoclonal antibodies, anti-HIV neutralizing antibodies, for therapeutic purposes using adeno-associated virus, AAV vector, for the delivery. In the course of the presentation, I will present evidence for an actual cure of a SHIV-infected monkey using this approach, a monkey that I hope by the end of the talk you will be calling the Miami monkey. Antibody-based strategies are made possible by the incredible array of monoclonal antibodies with potent neutralizing activity against a broad range of HIV-1 isolates that have been isolated and characterized over the last several years. As we heard in the last talk, one could consider delivering these antibodies by passive administration over the short term. And I think if we're looking long-term for worldwide use for prevention purposes over the course of decades and therapeutic purposes over the course of decades, passive infusion of antibodies suffers from a number of drawbacks, including it being very expensive and anticipated compliance issues. So my laboratory has focused on trying to achieve long-term delivery using vectors where the possibility of lifelong protection can be achieved from a single, relatively inexpensive injection. Adeno-associated virus, AAV, is ideally suited for such a long-term delivery vehicle. It's ideal in many respects. First and importantly, the only protein expressed from AAV vector comes from the transgene that one puts into it. So there are no AAV proteins that could be viewed as foreign expressed by the AAV vector. So as long as the protein that comes from the transgene that you put into it is viewed as self, is not viewed as foreign, long-term expression can be achieved. The most AAV vector deliveries are given into the muscle, into muscle cells. Muscle cells essentially don't turn over. They live forever. And if the protein product being made by that muscle cell is viewed as self, those muscle cells can serve as factories for production over the course of years and decades. And I'll show you one slide to illustrate that long-term expression that can be achieved. There is an outstanding safety record using AAV in human gene therapy trials. In fact, AAV has become the vector of choice in replacing protein products missing in certain hereditary disorders. Safety record is outstanding, and there is essentially no integration of the AAV vector DNA into the host genome sequences. The vector design my lab is using is basically a bicistronic construct. Well, not really bicistronic, but the coding capacity of AAV vector is about 4.9 kb, and one can just about fit in coding sequences for an entire heavy chain and entire light chain. We've used a variety of promoters. What I'm going to talk about today is the CMV promoter with the heavy chain sequences and light chain sequences made from a single transcript. And between the heavy chain and light chain sequences, there's a F2A self-cleaving peptide to create cleavage, and a furin cleavage recognition sequence that helps assure complete cleavage and also creates the exactly proper C-terminal amino acid at the end of the heavy chain. I talked about the ability to achieve long-term delivery of antibodies. This shows one of the first monkeys that we did with AAV delivery, a monkey numbered 8405. It received AAV making an anti-SIV authentic rhesus monkey monoclonal antibody called 5L7. Single inoculation on day 0 into the muscle cell, and our last measurement was well over 3.5 years later, and this monkey has been making constant levels of approximately 250 micrograms per ml of this antibody for over 3.5 years. This monkey has, and again, that's not to be too repetitive, a single inoculation, not repeated infusions, single inoculation, maybe $50 worth of AAV vector. This monkey has withstood 6 intravenous challenges with the difficult virus to protect against SIV MAC239, including challenges with 10 IV infectious doses of SIV MAC239. Okay, so I'm going to focus on a therapy trial. We enrolled 4 monkeys in this therapy trial, inoculated with SHIV-AD8. We chose SHIV-AD8 because it's a clone virus, and it results in rather consistent chronic phase viral loads, which is, if you have to pay for the monkeys, that's an important consideration. And these 4 monkeys were infected for 86 weeks with this SHIV-AD8 prior to initiating the AAV therapy. And these monkeys never were on antiviral drug treatment at any time point, either during the SHIV infection phase, around the AAV administration, or after the AAV administration. No antiviral drugs. You can see chronic phase viral loads are mostly in the 10 to the 3, 10 to the 5 range, pretty much what is seen in HIV-1 infected people not on antiviral drug treatment. So we had AAV vectors to deliver 3 monoclonal antibodies, 10E8, 3BNC117, and 101074. 10E8 is a GP41 MPER monoclonal antibody. 3BNC117 you heard about in Rebecca's talk is an anti-CD4 binding site antibody, and 101074 is a C2V3 carbohydrate-dependent monoclonal antibody. In all cases, we resized these monoclonal antibodies. That is, we converted the constant regions, these are human antibodies isolated from humans, so we resized them by converting the constant regions to authentic rhesus monkey sequences. Furthermore, we used IgG1 versions of these antibodies, and we did that intentionally to maximize the FC receptor-mediated activities, particularly antibody-dependent cellular cytotoxicity. As I will show you, we successfully delivered 10E8 to 0 of the 4 monkeys, and I'm going to give you some details on this. We successfully delivered 3BNC117 to 1 of the 4 monkeys and 101074 to 3 of the 4 monkeys. And the one monkey that was successfully delivered 3BNC117 was also successfully delivered 10-10-74. Here I show the data for that. The left column shows the levels of delivered antibody over time, with week 0 here being the week 86 when we administered AAV. 10E8, levels of delivered 10E8 here, levels of 3BNC117 here, and levels of 10-10-74 here. You can see, although you can't see it on this scale, there are little blips of 10E8 delivered at week 2 and week 4, but coincident with the appearance of a strong antibody response to 10E8, those levels of delivered 10E8 decline to lower undetectable levels. Strong correlation, strong anti-antis, very poor deliverability of the antibody. With 3BNC117, you can see good delivery of one of the monoclonals and in one of the 4 animals, and that is the animal with little or no anti-anti response. This is a pattern we've seen over and over again with a variety of different rhesus monkey antibodies and human antibodies, and anti-anti response severely limiting the consistency of antibody delivery. With 10-10-74, I should say this is not the first time we've used these antibodies. We've seen these sorts of patterns, 10E8 being the most difficult to deliver, 10-10-74 being the easiest to deliver, and reasonable delivery of 3 of the 4 animals of the 10-10-74. We've published on this. I think this is a 2015 paper where we've detailed the characteristics of the anti-antibody response. And these anti-antis are directed exclusively or almost exclusively to the variable domains. That is, they are anti-idiotype. So they are not reacting with the constant regions, but they are reacting to the variable regions. And it can be a bit surprising to think of an antibody being viewed as foreign, but I think you have to remember that there's a complex checks and balances system that allows some antibodies in your body to be released into circulation and others don't get out. It's a complex checks and balances system. And the antibodies that you're making are different than the antibodies that I'm making. So it's perhaps not so surprising if you take my antibodies and put them in you. You've not seen those variable domains before. They've not passed through the checks and balances system, and they can be viewed as foreign. Already covered that. Okay, now I'm going to focus on the one monkey, monkey 2438, where we did successfully deliver 2 of the antibodies, 3BNC117 and 10-10-74. And the levels of delivered antibody over time, so this week 0 is week 86 after the SHIV infection, and as a function of time, this is out 9 months. We've gone considerably longer. In blue is the levels of 10-10-74 antibody that we delivered, and in green, the levels of 3BNC117. So essentially, these 2 antibodies have been delivered at 75 to 150 micrograms per ml for now more than 18 months. 10E8, you can see peters out. These are the viral loads in that monkey. So the chronic phase viral load set point varied between 10,000 and 20,000 copies of viral RNA per ml of plasma. And on the day of AAV administration at week 86, it was 11,000 copies of viral RNA per ml of plasma. This is just about what the average is in HIV-1 infected people who have not on antiviral therapy. You can see a rather abrupt decline in viral loads, and viral loads below the limit of detection for 18 successive measurements over a 17-month period without any blips. I'm not sure this contains all of the time points. You can try to count them, but it's the latest is 18 time points, 18 months, 17 measurements, all below the limit of detection of 15 copies per ml in the hands of Jeff Lifson. It was surprising to us, and it's surprising to others in the field who know of these results, that virus resistant to these two antibodies did not emerge in this monkey. You know how resilient HIV and SIV are. They can change, adapt, and quickly select for variants that have a fitness advantage for replication. We, the two postdocs who were doing this work in my lab and myself discussed this for some period, and we came up with a sort of our theory, our favored hypothesis for why we did not see the emergence of resistant virus. And then we went to the literature and found that that explanation was already out there. So Michel Nussenzweig and his group published in Journal of Experimental Medicine in 2014 results where they selected for HIV-1 resistant to these two antibodies using humanized SCID mice to which they administered these two antibodies. So there's no immune response of the humanized mice. They delivered the two antibodies. They selected for resistant variants. And it turns out that virus resistant to these two antibodies becomes an extreme tier 1 virus, a virus that's hammered by any antibody you throw at it. So the explanation here of what's going on is, yes, it's the two antibodies, but it's also the antibody response of the host, that the three of them are working together to not allow the emergence of virus that's resistant and not allowing a resurgence of viral replication. Next, we looked at antibodies to the P27 gag. And I hopefully have enough slides here to explain why that's relevant. So this is week 86, the time of AAV administration. This is week 0, the time of SHIV-AD8 infection. And you can see over the subsequent 86 weeks, strong levels of antibodies to P27 gag. And following the administration of the suppressive AAV monoclonal antibodies, there's been a more than 20-fold decline in the levels of anti-P27 gag antibodies. What can we compare that to? If one looks at anti-P24 antibodies, let's look at these two sets. So this grouping is individuals' levels of anti-P27 gag antibodies prior to going on antiviral drug therapy. And this is the same 10 individuals 4 to 5 years after going on effective antiviral therapy. And there may be some slight declines in a few individuals, but there's essentially little or no decline in the levels of anti-P27 antibodies. Okay? So the decline we're seeing is unusual and not representative of what happens when a person in the chronic phase of HIV-1 infection goes on antiviral drugs. Let's look at the Berlin patient, Timothy Brown, for comparison. So these are Western blots. This is the original New England Journal paper. Lane 2 is a Western blot from the Berlin patient taken 14 days before his stem cell transplantation. And you can see strong bands to GAG, P24, GAG, P17. And lane 3 is 625 days. That is 1 year and 9 months, something like that. 21 months after the stem cell transplantation. And you can see still some weak bands, but a dramatic decline in the anti-P24 GAG antibodies. And back to the same paper, they also included samples from the Berlin patient 4 to 5 years after his stem cell transplantation. And by then, they had declined to background levels indistinguishable from uninfected people. So the nature of the anti-P27 antibody decline is more reflective of what has been seen in the Berlin patient than what is seen on humans on effective antiviral drug treatment. Antibodies to envelope also reflect those two comparison groups, Timothy Ray Brown and humans on antiviral drugs, in that there has been a decline, but it's not as dramatic, and it's much the levels of antibodies, for whatever reason, are much more persistent. All of our attempts to recover SHIV from peripheral blood of this animal following the AAV administration have failed. Here I show our recovery attempt at week, I think it's week 62, yeah, week 62, following the AAV administration. We obtained we did a large blood draw for a monkey and obtained 180 million PBMC. We used magnetic beads to deplete CD8 cells, which left us with 100 million cells. We put 50 million on the 221 cell line, which is permissive. It's a rhesus monkey continuously growing cell line that's permissive for the SHIV-88 that we used. And we put 50 million co-cultivated with PHA-activated PBMC from a naive donor in the presence of IL-2. And both these recovery attempts failed, as has all other attempts to recover virus from this monkey. More recently, around week 80, we've done a similar bleeding, similar isolation. This time we included 2 SHIV-infected monkeys as positive controls. We readily recovered virus from these 2 SHIV-infected monkeys as positive controls using both of these co-cultivation conditions, including the one monkey that had marginal viral loads, a copy number of 25 copies of viral RNA per ml of plasma only. Again, we readily recovered virus with SHIV appearing in the cultures first around day 8, day 9. We've kept the culture from the monkey 2438 for 28 days and have not recovered virus again. DNA, RNA, I'm not sure we fully understand yet in terms of reservoirs, and we're continuing to study that. But RNA reservoirs, in terms of numbers of RNA copies, this is GAG in both cases. RNA and DNA is copies of GAG using GAG primers. Declined very early, about 2.5 logs, but there have been detectable levels of GAG RNA continuously detectable out to the present time. To what extent, our best guess is that this represents defective proviral genomes, not competent for replication, but that those investigations are in progress. And similarly, there is a decline, but not to zero, in the levels of DNA reservoirs. And finally, in terms of data on this monkey, and most importantly, we've made 2 attempts at adoptive transfer. So here I show the first attempt at the adoptive transfer, where we removed a lymph node from 2438 at 74 weeks after the AAV administration. We made a single-cell suspension of the lymph node and took the entire amount, 38 million lymph node cells, and infused it intravenously into a naive recipient monkey, and the recipient monkey has not become infected from that adoptive transfer attempt. Second adoptive transfer attempt was initiated at 79 weeks after the AAV administration. We got Institutional Animal Use Committee approval to remove 3 entire clusters of lymph node from this one monkey on the same day. We took all the lymph nodes from these 3 clusters, made a single cell suspension, again, this time 139 million lymph node cells. We adoptively transferred those 139 million lymph node cells intravenously into a separate new naive rhesus monkey recipient, and again, that recipient did not become infected. So by the best available evidence that we can do, no one's going to do an adoptive transfer attempt in the Berlin patient, but by the best available data that we can muster, the Miami monkey 2438 has apparently been cured of its SHIV infection by the long-term administration of these 2 monoclonal antibodies. This is the Miami monkey. I've been told that he's quite happy and quite frisky. Yes, we've not submitted this for publication yet. Yes, the N is only 1. But I would make a couple of points on that. First, I would say it's 1 of 1. This is the only SHIV-infected monkey that we've delivered these 2 antibodies to, and and it appears to be an apparent cure. And secondly, I would make the point that the Berlin patient is also 1, a single example, and look at how results with the Berlin patient has changed the field's thinking, has greatly influenced science funding toward cure research and toward a cure. However, I certainly agree that it's important that we do more monkeys and see how consistently can we get this result and what parameters influence it. So we've now infected 12 more monkeys with SHIV-AD8 with the intentions of subsequently enrolling them in a similar therapy trial. Our current thinking is we're going to use 6 of the monkeys for AAV delivery of these 2 antibodies, 3BNC117 and 1074. And the other 6 will go on highly active antiretroviral therapy, so we can have a group in parallel that we can compare these parameters: ability to recover virus, anti-P27 gag antibodies, and depending on the results, in some monkeys, attempts at adoptive transfer. And it's important to note at this stage for this experiment that most of the effort in my lab in the last 2 years has actually been directed to alterations in AAV vector design and delivery conditions that can minimize the anti-anti problem and allow for, and maximize the consistency of antibody delivery. And I can tell you that I think we now have those vector design and delivery conditions. We certainly have them to consistently deliver our most problematic antibody, an antibody called 4L6. We can now consistently deliver that, and we're hoping that we can also consistently deliver these two antibodies to be able to increase the N substantially. Trials in humans. Monkeys are monkeys, humans are humans. Certainly, I'm a strong advocate for monkey experiments to inform and guide development of concepts for use in people. Personally, I feel that we should not wait, that this is ready now. We don't know to what extent anti-antis are going to be a problem in humans, for example. And I have been sort of stepping out of my box of what I've done historically and trying to interest the proper parties in getting this to human testing in HIV-infected people. Okay, that's it. I want to several people I want to thank in particular. So almost all of the work has been done by two people in my lab, Sebastian Fuchs, part of our joint graduate student training program with the university in Germany, and Chema Martinez Navio, a postdoc from Spain. All of the plasmid DNAs for AAV vector have been designed and constructed by Sebastian in my lab, but the actual vector production and quality control has been done by Guangping Gao at UMass Medical School. The monkeys have been held at Wisconsin, and I thank Wendy Newton, Ava Rakaz, and their teams for taking care of the monkeys and sampling the monkeys. Viral loads and genetic analysis are being done, Jeff Lifson and Brandon Keel at Frederick. I thank Malcolm for providing the SHIV-88 clone prior to publication, and I particularly thank Michelle Nussensweig for providing the antibodies and being supportive of these studies every step along the way. Okay, thank you very much.